All right, time to go teach. Hello and welcome to this, our second episode dealing with photosynthesis. As we continue our tour of the cell, we are just going to continue into the second stage of photosynthesis today, which is the Calvin cycle. So we are going to look at that stage, which is the light-independent reactions. We're going to identify the products and the reactants involved. And lastly, we are going to summarize all of the events of photosynthesis to make sure that we understand what's really important about this process. So remember, the light-dependent reactions, they occurred on the thylakoid membrane, and they used water, split it open using the energy from light. It also used an adenosine diphosphate, a phosphate, and an NADP+. All of those were used to actually end up releasing oxygen and building our NADPH and our ATP. And the NADPH and the ATP were in the stroma now. They have been built in the stroma, and they are energy-carrying molecules. They are going to be able to transport the energy from one location to another location. And in this case, we are just transporting the energy from the thylakoid membrane to the stroma, which is not very far away, but that's what we're doing. The energy went from the light-dependent reactions that was captured from light and now is going to be carried in these molecules to power the Calvin cycle. And in fact, it was going to take quite a few ATPs and quite a few NADPHs to power the Calvin cycle. And we're going to look at how this process works. Some people show this process as being a circle. It is a cycle. That's why it's called a Calvin cycle, because the end product of the Calvin cycle is also what is used to start the Calvin cycle. And so that's why we refer to it as a cycle. The end and the beginning are exactly the same molecule. It actually takes many reactions to get from the beginning to the end. I'm going to show it in sort of a linear way because I don't want you to think that this actually just takes place in a specific location within the stroma. It takes place throughout the entirety of the stroma. These molecules are just going to get broken down and built up in different reactions in the stroma. It's going to use that NADPH and the ATP to actually power those different reactions, to provide the energy that is required for those different reactions to take place, to provide the activation energy for them. So first, CO2 is going to diffuse into our stroma. And in this case, I'm actually going to go through 3 different Calvin cycles. So that's why I brought in 3 different molecules of carbon dioxide. So here are 3 different carbon dioxides. And there's also going to be another molecule called RUBP. It's a 5-carbon molecule that exists in the stroma. It is going to be there. It will be present in the stroma. This is naturally in the stroma. RUBP stands for ribulose bisphosphate, and it is a 5-carbon molecule. And now what I'm going to do is I'm going to remove those oxygens from the carbon dioxide because the only thing we're going to pay attention to is the number of carbons. We are not going to pay attention to the oxygens. We're not going to pay attention to the hydrogens. We are going to keep this as simple as possible to just track the carbons. That is the easiest way to understand the Calvin cycle and what's taking place. So, removing our oxygens, here we go. First, carbon dioxide is actually going to bond to the RUBP using an enzyme called ribisco. And ribisco is a really useful enzyme because that starts our Calvin cycle. We need that. So now that the carbon dioxide has bonded to the RUBP, we now have a 6-carbon molecule. And that 6-carbon molecule is going to very rapidly break down into 3-carbon molecules. It is an unstable 6-carbon molecules. So it breaks right in the center, and we end up with 2 3-carbon molecules. So now, those 3-carbon molecules are going to get converted a little bit, and when they do, they are going to get converted into a molecule called glyceraldehyde 3-phosphate, or G3P. That molecule is extremely important. We had to use up ATP and NADPH for each cycle. It was very costly for this production of the glyceraldehyde 3-phosphate. But the reason why it's important to build the glyceraldehyde 3-phosphate is because it is a precursor to glucose. You see, it takes 2 glyceraldehyde 3-phosphates for the cell to produce glucose, which is a 6-carbon molecule. And now we have 6 glyceraldehyde 3-phosphates. But if the cell used every single one of these glyceraldehyde 3-phosphates to build glucose, it would have a problem. It would have nothing left to start the Calvin cycle again. So it needs to build more RUBP. And that's why the cell is actually going to only keep one of these glyceraldehyde 3-phosphates to use to build glucose. The other glyceraldehyde 3-phosphates are going to be used, and the rest of ATP will be used for this process to rebuild RUBP. So for 3 carbon dioxides going through 3 different Calvin cycles, you end up producing 1 glyceraldehyde 3-phosphate that is required to produce glucose. And if it takes 3 carbon dioxides to produce 1 glyceraldehyde 3-phosphate, and it takes 2 glyceraldehyde 3-phosphates to produce 1 glucose, then very clearly it takes 6 carbon dioxides going through the Calvin cycles to produce 2 glyceraldehyde 3-phosphates to produce 1 glucose. And that is the way that it works out. That is how the Calvin cycle works. So photosynthesis, very simply, has a process where light starts it all. Light starts the light-independent reactions which occur on the thylakoid membrane. Why in the world do we have a chemical reaction that just says 6 CO2 plus 6 H2O yields 1 glucose and 6 oxygens? Why is that the chemical reaction when all this other stuff is put in and all of this other stuff comes out? Like, we put in an adenosine diphosphate into the ATP synthase. We also put an phosphate in there so that way it can produce ATP. That's all true, but the thing that we can see here is that, well, ATP, it's both an input and an output. And ADP and the P, they're also an input and an output, as is the NADPH and the NADP+. Because the energy-carrying molecules are on both the product and the reactant side, we don't put them in the chemical reaction. We don't include them because they would be on both sides. So even though they are used up and built in this chemical process, we don't include them in the chemical reaction. They are left out for simplicity's sake. And that's why our chemical reaction is just the 6 carbon dioxides plus the 6 waters yields 6 oxygens plus 1 glucose. So photosynthesis, in summary, it just converts that light energy to chemical energy. And it ends up building a molecule that we can end up storing. The light-dependent reactions, that produces the ATP and the NADPH, just to carry the energy from the thylakoid membrane just as far as the stroma. Not very far at all, but it's going to take that chemical energy, that little distance, so that way that those different chemical reactions can take place so that G3P can be produced to make glucose. And now the plant cell has energy, chemical energy, that it has stored. And glucose, well, that is a primary energy storage molecule for life. And that's it for photosynthesis. Till next time, be awesome, stay awesome.